Hello guys, as you all know that we have started the physiology of GIT. In that we have studied about the secretions, we have studied about the hormones that are secreted through the GIT tract, and after that we have started the absorption and digestion of that what are the substances that are absorbed and digested inside the GIT tract. So in that we have studied about the carbohydrates. We have studied about the lipids, how they are absorbed and how they are digested. So today we are going to start about the new component, that is proteins. That how proteins are digested, and after digestion how they are absorbed into the body. So we all know that in diet proteins are polypeptides. They are the combinations of different types of amino acids, and when they combine together, they form a polysaccharides. So, in mouth, for protein digestions, we have no enzyme. That is why, whenever we take any protein, we do not feel the taste inside the mouth because we don't have any enzyme for the protein digestion. From the mouth, the protein moves to the stomach. So in stomach, for protein digestions, we have one enzymes, that is pepsin. So pepsin is secreted by the stomach that helps in the digestion of the proteins. After that, we have exocrine pancreas. We know that pancreas we're divided into two parts: the endocrine pancreas and the exocrine pancreas. So the endocrine pancreas, it almost related to the release of the insulin, mostly, but the exocrine pancreas, it is concerned with the digestion or secretion of different types of enzymes that helps in the digestion of the food. So among the exocrine pancreas secretions, we have trypsinogen, we have chemotrypsinogen, we have procarboxypeptidases. So all these exocrine pancreas, they are secreted inactive. They are secreted in inactive form. As they are inactive, so we call them as zymogen. Why they are secreted inactive? Because to prevent the autodigestion of the pancreas, they are so powerful enzymes. If they secretes actively, they will eat pancreas itself. So in order to prevent the autodigestion of the pancreas secrete these enzymes as an active form. Now, when these enzymes come to the intestine, so in intestine, suppose we take the trypsinogen. So this trypsinogen, it convert into trypsin. This trypsin is the active form of this trypsinogen. So this conversion from trypsinogen, an inactive form to an active form trypsin, this is done by an intestinal enzyme which is called enterokinase. So enterokinase actually what it did? It did, or it convert the inactive form of trypsinogen to an active form trypsin. After that, it will do the digestion of the pancreas. So now we divide these enzymes into two categories. How? Look at, we consider is trypsinogen and chemotrypsinogen. Trypsinogen and chemotrypsinogen, they act at the anterior side. We already know that proteins are in polypeptide. Polypeptide means that they are consist of so many peptides bond. So these trypsinogen and chemotrypsin, they act inside at interior end of this polypeptides. That is why they are called endopeptidases. They are called endopeptidases because they act interior to this polypeptides. And procarboxypeptidases, they are called exopeptidases because they act at the outside, or you can say that they act at the sides of this polypeptides. So that is why they are called exopeptidases. Now when this polypeptide, it comes to intestine, it converts into disaccharide or trisaccharides. These polypeptides protein, they will convert into disaccharide or trisaccharides. So how these proteins are then absorbed into the body? Now look at. Now, as we have discussed that the polypeptide form of the protein, they are digested into dipeptides or tripeptides by different types of the enzymes that are secreted from the stomach as well as from the exocrine pancreas. Now, it is proved that we have formed dipeptides and tripeptides. Now, consider this as an intestinal cell. This is an intestinal cell because the digestions further and the absorption that takes place there in the intestinal cell. So, for instance, this is an intestinal cell. These intestinal cells come nearest to blood vessels. This is a basolateral sides and this is a luminal sides. The sides which near to the vessels we call that basolateral, and that is the luminal side. So in the lumen, the dipeptides and tripeptides, they are present. What we have to do? We have to do that these dipeptides or tripeptides, they have to transport it into the intestinal cell for their further digestion. So here we have a channel which is called PEP-T, peptidase transporter or peptide transporter. So what does it? This channel, it bring dipeptides or tripeptides into the intestinal cell, and in response to the dipeptides or tripeptides, this channel also bring one hydrogen ion inside the intestinal cell. Now, as we know that hydrogen ion is related to the acidity, so as when it comes inside the cell, it disturbs the internal environment of the intestinal cells. So in order to maintain this internal environment, what we have to do, this cell throw this hydrogen outside. It should throw this hydrogen outside in order to maintain its internal environment normal. So what it does, we have a channel which is called sodium hydrogen exchanger. What this actually do, this channel throw hydrogen ion from inside of the cell to the lumen, and in response of that hydrogen ion, this channel throw one sodium ion in response to hydrogen ion inside to this cell, intestinal cell. So now what we have inside the intestinal cell, we have enzymes that are called peptidases. What actually these peptidases do, they digest this dipeptides or tripeptides into amino acids. They are digested into the amino acids, and when they are digested into the amino acids, these are the amino acids which should be transferred into the blood vessels, and these are the amino acids which should be absorbed from the intestinal cells. So we have a channel which is called basolateral amino acid transporters. This channel throw the amino acid into the blood vessels where they get absorbed. So this was all about the protein absorption and protein digestions. Let's revise it a little bit. That we have studied that protein are polypeptides. They should be converted into dipeptides or tripeptides in order to further digestions, and in order to digest, and in order to absorb inside the body. What actually did? So these dipeptides or tripeptides, they are transported into the intestinal cells. There we have peptidases. These peptidases, what actually they do? They do the digestion of the dipeptides or tripeptides into amino acids, and these amino acids are transported into the basolateral vessels by amino acid transporters, and they are get absorbed. Now after protein digestion and absorption, we should know a little bit about that how vitamins are absorbed. So we have two types of vitamins. Number one, we have fat soluble vitamins, and number two, we have water soluble vitamins. So fat soluble vitamins, they are vitamin A, vitamin D, vitamin E, and vitamin K. So how they are absorbed into the body? We already know about a structure called micelles. What actually they do? They are incorporated into a structure called micelle, and after when they are incorporated into micelles, they are get absorbed into the mucosal epithelial cells. They are absorbed into the mucosal epithelial cells. This is how the fat soluble vitamins are absorbed into the body. They are incorporated into a structure called micelles. And what are the water soluble vitamins? So the water soluble vitamins are called B complex. They are absorbed by using a sodium dependent co-transporter, except how they are absorbed? Sodium dependent co-transporter, except what? Folate and vitamin B12. They are sodium independent processes. So the folate absorbed in the duodenum, as they are independent of the sodium. So folate is absorbed in the duodenum, and vitamin B12, it gets absorbed in the terminal ileum. So this was all about a short story about the vitamin absorption, that how they are absorbed inside the body. Now remember, we have already discussed about it, that our GIT, it secrete almost 9 liter secretion per day. Out of that, 8.8 liter is totally absorbed, and zero point 0.2 is secreted inside the stool. Remember that maximum water or fluid absorption through the GIT tract, it occur at the level of the duodenum. Maximum fluid or water absorption, it takes place at the level of the duodenum. Okay, so now we will discuss a little bit about iron absorption, that how iron is absorbed into the body. So iron absorption occur in the duodenum. It is absorbed inside the duodenum. So in diet, iron is usually present in ferric form, Fe plus 3, but usually it is absorbed into the cell in the form of ferrous iron, or it is absorbed in ferrous form. So let's suppose this is a duodenal cell which come near a blood vessels, and we have a luminal site. So inside the lumen, what we have? We have ferric iron which came in diet. So this ferric iron, it gets converted into ferrous iron by removal of one iron by the enzyme called ferrireductase. Ferrireductase, it convert the ferric iron into the ferrous iron. So inside these cells we have two transporter. One is called HCP and the other is called DMT1. So this HCP is called heme carrier protein. What it does? It from the lumen, this heme carrier protein, it bring heme inside these cells. And DMT1, it is called dimetal transporter. What actually it does? It transport this ferrous iron into the cell of the duodenum. So HCP, it transport the heme inside. Fe plus 2, it is transported by the DMT1, and inside the cells the ferrous iron again it converted into ferric iron. It again converted into a ferric iron. So on basolateral side, now what we have done, the ferric iron converted into ferrous iron. The ferrous iron, it get inside these cells, and this ferrous iron again it converted into a ferric iron. Now on this basolateral sides we have a transporter which is called ferroportin. This transporter is called ferroportin. protein which transport this ferrous ion and convert it into ferric ion by an enzyme called hephaestin. Here, this ferrous ion, it convert again into a ferric ion, and this conversion of ferrous ion into a ferric ion by an enzyme called hephaestin. Let me write here, hephaestin. This, what this enzyme do, this convert this ferrous ion into a ferric ion. Now Fe3, the ferric ion, it come to the blood vessels, and inside the blood vessels it is combined with transferrin, Fe plus 3. It comes to the blood vessels. Here it combined with what? With transferrin. And when it combined with transferrin, after that it get absorbed into the body. So remember, iron is stored in the form of ferritin inside the body, in the form of ferritin. Okay, so what we have done, it is stored in the, or it gets absorbed in the duodenum. In diet we have it is in the ferric form, and we should absorb it inside the cells in the form of ferrous. So in the lumen, ferrireductase, it convert the ferric ion into the ferrous ion, and DMT1, dimetyl transporter, it convert this ferrous ion inside the cell. It is converted by hephaestin into ferric ion, and this ferroftin, it is a transporter. We transport this ferric ion to the blood vessels. In the blood vessels, it is combined with hephaestin, and after that it get absorbed inside the body. Now after that we will discuss about the what are those substances that increase iron absorption inside the body, and what are those substances that decrease the iron absorption inside the body. So the substances that the substances that increase iron absorption is vitamin C or ascorbic acid. It increase iron absorption. Substances like tannins, platelets, oxalates, they decrease iron absorption inside the body. This tannins it is present inside the tea, and what these two substances actually do, the platelets and oxalates, they form the complexes, the complex within iron, and decrease its absorption inside the body. After that, we should also learn about the regulation of iron absorption, that how the absorption of iron it is regulated. So iron absorption, it is regulated by an important molecule called hepcidin. It is secreted by liver. Hepcidin is secreted by the liver. In the presence of hepcidin, what actually changes occur when there is hepcidin? Ferroportin, we know that it is a transporter that convert the ferrous ion into the ferric ion by an enzyme hephaestin. So ferroportin, in the presence of hepcidin, it undergo internalization. It undergo internalization and get destroyed, due to which iron absorption decreases. When ferroportin it is destroyed, when there occurs a changes inside this, so obviously it is a transporter of the iron into the blood vessels. After that, the iron will not be transported into blood vessels. In order, what does it did? It decreases the absorption of the iron inside the body. So what does it mean? It actually mean that hepcidin, it is a negative regulator, means that it reduces the absorption of the iron. Now whenever there is any mutation inside the hepcidin, what will be the result? When there is a mutation, obviously there will be a more absorption of the iron, because the ferroportin will not be regulated properly, and it will absorb more iron. So what will be the end result? The end result will always be characterized by iron overload inside the blood, and that iron overload is called hemochromatosis. So this was all about the digestion and absorption of different types of molecules that we have discussed. In the first, we have discussed about the carbohydrates and lipids in the first part of this lecture, and in this second part we have discussed about the protein digestion and absorption. We have discussed about the vitamin absorption, the iron absorption, its regulations. So this module of absorption and digestion, it comes to an end, and after this we will discuss about the last module in this GIT physiology, which is the mortality inside the GIT tract. So if you are new on our channel, please subscribe to our channel. Thank you so much.